Welcome to Week 4, Chapter 9. In this chapter, we're going to go through some similarities and differences between DNA and RNA. We'll be looking at the processes of transcription and translation, which is where we take the DNA information, transcribe it into RNA information, and then translate it into polypeptide information. Additionally, we'll actually go through and translate a messenger RNA transcript. So let's get started. On this slide, what we see is a comparison of a nucleotide from RNA compared to a nucleotide from DNA. If you recall, nucleotides are the building blocks of nucleic acids, and each nucleotide is made up of three basic structures. Three phosphate groups, as you can see here, are common to both a DNA nucleotide and an RNA nucleotide. A 5-carbon sugar is central in both nucleotides. In RNA, the 5-carbon sugar at carbon number 2, that's what this means, has a hydroxyl group. And down here in the DNA, the 5-carbon sugar only has a hydrogen. That's why this is called deoxyribose, because deoxy means without hydrogen. So if you compare the carbon 2 on the ribose to the carbon 2 on the deoxyribose, you can see deoxyribose is without an oxygen. Also similar to both RNA and DNA nucleotides are the nitrogenous bases, in the sense that of the 4 nitrogenous bases present in DNA, which is adenine, thymine, cytosine, and guanine, those are also present in RNA, except uracil takes the place of the thymine. So, just to summarize, nucleotides in both DNA and RNA are made up of a nitrogenous base, a 5-carbon sugar, and at least one phosphate group. When the nucleotide is by itself and not attached to another nucleotide, it will contain 3 phosphate groups. When it is attached in a string with other nucleotides, like when you're forming a DNA molecule or when you're forming an RNA molecule, it will only have 1 phosphate group. So now let's compare the entire DNA structure with the entire RNA structure. Recall from the last slide that we looked at, common to both are nucleotides. And as I stated, when you join nucleotides together, you lose 2 of those 3 phosphate groups because they basically provide the energy for that joining. And that joining is called a phosphodiester bond. So if we look on the left in this image, we can see the DNA, which is a double-stranded molecule. Here is one of the strands. This copper-colored ribbon is where the sugars and phosphates are. That's called the sugar-phosphate backbone. And these blue bars, those are the nitrogenous bases. So you can see DNA is made up of 2 strands, so it's called a double-stranded molecule. The backbones of both strands of DNA are identical. The sugar is deoxyribose, and then 1 phosphate alternates between those sugars. What makes the coding in the DNA different are the nitrogenous bases, which are represented by these blue bars here. And over to the left, we can see each bar is either an adenine, a guanine, a cytosine, or a thymine. Notice that the nitrogenous bases are either 2 rings, such as we see here with adenine and guanine. 2-ringed nitrogenous bases are called purines, or they are single-ringed, and single-ringed nitrogenous bases are called pyrimidines. And in DNA, those are cytosine and thymine. So when we join double strands of DNA together, and we have nitrogenous bases bonding through hydrogen bonding across the middle, holding those 2 strands together, what we're pairing are 1 purine with 1 pyrimidine, always, so that we always only have 3 rings two from a purine and one from a pyrimidine between those two sugar phosphate backbones. So adenine always pairs with thymine, and cytosine always pairs with guanine. Those are called complementary base pairings. So now let's go over here and compare RNA to the DNA. We can see a similarity in that we have a sugar phosphate backbone. Recall a uniqueness with the RNA is instead of having deoxyribose as your 5-carbon sugar, you have ribose as your 5-carbon sugar. Notice another difference with the RNA is it's single-stranded, not double-stranded. These bars are representing the nitrogenous bases of RNA. There are two types of nitrogenous bases in RNA, just as we saw in DNA. The two rings are called purines. The single ringed are called pyrimidines. Please notice that we have an adenine in RNA as well as a guanine, so our purines are the same in DNA and RNA. We also have a cytosine in RNA as we do in DNA, but we do have a unique exception here with uracil. Uracil is a pyrimidine only found in RNA, just as thymine is a pyrimidine only found in DNA. So why have two types of nucleic acids? Well, basically the DNA information stays within the nucleus, and on one chromosome you have one double-stranded DNA molecule, and that one double-stranded DNA molecule has multiple genes on it, and at any one given period of time in a cell, not every gene on that double-stranded DNA molecule is going to need to be expressed or basically make a product. So since you only need to turn on or express certain genes at certain times, your DNA is kind of like a storehouse of knowledge. It's like the hard drive of your computer, and you don't want it out in a hostile environment such as the cytoplasm of the cell could be. And the cytoplasm of the cell is where we're going to have the translation process occur, which is where we make a polypeptide from our genetic information. So we don't want to have the DNA out there. One, because one chromosome contains multiple genes, and we're not going to need those multiple genes being expressed at the same time. And two, that's a hostile environment, and we need to keep our DNA protected inside of the nucleus. So for us to express the DNA, which basically means make a protein from it, we're going to transcribe the DNA language into RNA language. And the only section of the DNA language that we're going to transcribe is the gene. So the part that we want to express in essence. That RNA that is transcribed from the DNA will then leave the nucleus and go into the cytoplasm, where it can then be translated into amino acid language, which makes a polypeptide, which is also known as a protein.